and welcome again. Today we will discuss one more important topic, that is the plasma membrane and its structure and functions. As we know that plasma membrane, also called as cell membrane or plasma lemma, is a boundary that separates the living cell from its surroundings. The plasma membrane is selectively permeable, allowing only certain substances to cross it more easily than the others. The plasma membrane can be studied under three sections. 1. Membrane composition, 2. Membrane structure, and 3. Membrane functions. First of all, we will take up the membrane composition. The main components of the cell membrane are lipids, proteins, and carbohydrates. Lipids are a broad group of naturally occurring molecules which includes fats, waxes, sterols, fat-soluble vitamins, monoglycerides, diglycerides, and many other molecules. The lipid composition of the cell membrane includes phospholipids, sterols, and glycolipids. Phospholipids are the major structural components of the plasma membrane. They are also the main lipids found in the plasma membrane. Phospholipids are amphipathic molecules containing hydrophobic and hydrophilic regions. When placed in water, phospholipids self-assemble into micelles or bilayers, structures that exclude water from the hydrophobic regions while keeping the hydrophilic heads in contact with water. These specific properties allow phospholipids to play a major role in the phospholipid bilayer. Lipid bilayers occur when hydrophobic tails line up against one another, forming a membrane with hydrophilic heads on both sides facing the water. Two important properties of phospholipids are critical to the membrane function. First, the structure of phospholipids is responsible for the basic membrane function as barriers between the two aqueous compartments. Because the interior of the phospholipid bilayer is occupied by the hydrophobic tails, the membrane is impermeable to ions, including the most biological molecules. Second, the naturally occurring bilayers of phospholipids are viscous fluids, not solids. The fatty acids of most natural phospholipids have one or more double bonds which introduce kinks into the hydrocarbon chains and make them difficult to pack together. The long hydrocarbon chains of fatty acids are therefore free to move freely in the membrane, so the membrane itself is soft and flexible. In addition, both phospholipids and proteins are free to move within the membrane, a property that is critical to most membrane functions. Another component of lipids found in the plasma membrane are sterols. The steroids are naturally occurring or synthetic fat-soluble organic compounds based on a molecule with 4 fused carbon rings. This family of lipids includes many animal hormones and cholesterol. Cholesterol is a waxy steroid metabolite found in the cell membranes of animals. It is not found in the plant cell membranes. Cholesterol helps stabilize the phospholipids. Another class of compounds that is present in the plasma membrane are proteins. A membrane is a collage of proteins embedded in the phospholipid bilayer. Proteins determine most of the cell membrane's important functions. Two major populations of membrane proteins are peripheral proteins and integral proteins, also called as extrinsic and intrinsic proteins respectively. Peripheral proteins are bound to the surface of the membrane. Since they are superficially located, they can be easily removed from the membrane. On the other hand, integral proteins are tightly held. They penetrate the hydrophobic core of the membrane and are therefore difficult to remove. Like the lipid molecules, the integral proteins have hydrophobic and hydrophilic regions. The polar regions protrude from the surface and non-polar regions remain embedded in the hydrophobic interior of the membrane. The hydrophobic region of an integral protein consists of one or more stretches of non-polar amino acids coiled into alpha helices. Some smaller integral proteins partially penetrate the membrane and are exposed on one surface only. Many of the integral proteins are transmembrane proteins which completely span the membrane and project beyond the lipid bilayer on both sides. Transmembrane proteins can either pass the membrane one or multiple times. The former are referred to as single-pass proteins and the latter as multi-pass proteins. As a result of their structure, transmembrane proteins are the only class of proteins that can perform functions both on the outside and inside of the cell membrane. Many of these proteins are also believed to have channels through which water-soluble materials can pass. There are 6 major functions of membrane proteins: transport, enzymatic activity, signal transduction, cell-to-cell recognition, intracellular joining, and attachment to the cytoskeleton and extracellular matrix. The third class of compounds present in the plasma membrane are carbohydrates. Some proteins and lipids have carbohydrates attached on the extracellular side of the membrane, forming glycoproteins and glycolipids. These form a layer of carbohydrate chains on the outer side called glycocalyx. Carbohydrates are important for cell-to-cell recognition. Now we come to the second section of the plasma membrane, that is the membrane structure. Membranes have been chemically analyzed and found to be made up of proteins and lipids. Scientists studying the plasma membrane have reasoned that it must be a phospholipid bilayer. In 1935, Daniel and Dyson proposed the sandwich model for plasma membrane, in which the phospholipid layer lies between the 2 layers of globular proteins. Later studies found problems with this model, particularly with the placement of the membrane proteins that have hydrophilic and hydrophobic regions. In 1972, Singer and Nicholson proposed the fluid mosaic model for plasma membrane, in which the plasma membrane is a mosaic of proteins dispersed within the lipid bilayer with only the hydrophilic regions exposed to water. According to this model, the membranes are 2-dimensional solutions of oriented lipids and proteins. The scattered arrangement of protein molecules in the matrix of phospholipids gives the arrangement of a mosaic pattern, hence the name to the model. These membrane proteins are free to diffuse laterally in the membrane. An important property of plasma membranes is the asymmetry. Structurally, the lipid bilayer is asymmetrical. That is, the lipids and proteins in each of the 2 layers is different. So, the membranes have distinct inside and outside faces. The extracellular side of the membrane contains carbohydrates or oligosaccharides that distinguish the cell as self, and also contain the end of the integral proteins that interact with the signals from other cells and sense the extracellular environment. The inner side of the membrane anchors to the cytoskeleton and also contains the end of the integral proteins that relay signals received from the outside. Another important property of membranes is the fluidity. A membrane must be fluid in order to work properly. The fluidity of the membranes depends upon the structure of fatty acids and temperature. Membranes rich in unsaturated fatty acids are more fluid than those rich in saturated fatty acids. The right ratio of saturated and unsaturated fatty acids keeps the membrane fluid at any temperature that is conducive to life. In addition, the steroid cholesterol has different effects on membrane fluidity at different temperatures. At warm temperatures such as 37 degrees Celsius, the cholesterol restrains the movement of phospholipids. At cool temperatures, it maintains fluidity by preventing the tight packing. So, phospholipids in the bilayer can move freely within the membrane. Most of the lipids and some proteins drift laterally. Rarely does a molecule flip-flop transversely across the membrane. The third and last section of the plasma membrane is the membrane functions. While lipids are the fundamental structural elements of the membranes, proteins are responsible for carrying out specific functions. Most plasma membranes consist of approximately 50% protein and 50% lipid by weight, with the carbohydrate portions of the glycolipids and glycoproteins constituting about 5 to 10% of the membrane mass. A cell must exchange materials with its surroundings, a process controlled by plasma membrane. Plasma membranes are selectively permeable, therefore regulating the molecular traffic. The lipid bilayer is permeable to small non-polar molecules like carbon dioxide, oxygen, etc., but is impermeable to ions like sodium, potassium, chlorine, etc., and other polar molecules like protein, glucose, sugar, etc. The transport proteins enable membranes permeable to specific ions and other polar molecules including water. Thus, the selective permeability of a membrane depends on both the discriminating barrier of the lipid bilayer and specific transport proteins. The processes through which the substances can cross the membrane are passive transport, facilitated diffusion, active transport, and bulk flow. Passive transport is the diffusion across the membrane down the concentration gradient. In facilitated diffusion, a specific carrier protein helps the molecule to move across the membrane. Active transport is the pumping of solutes against the concentration gradient. Bulk flow involves exocytosis and endocytosis for the transport of large molecules. Passive diffusion is the net movement of materials from an area of high concentration to an area of low concentration. The difference in the concentration between the two areas is known as concentration gradient. The diffusion of materials continues until the gradient is eliminated, or in other words, the equilibrium is reached. This pathway does not require energy or carrier proteins and is taken by the molecules that are lipid soluble, like carbon dioxide, oxygen, nitrogen. Facilitated diffusion. In facilitated diffusion, a specific carrier protein with a central channel helps the molecules to move across the membrane. These specific carrier proteins that form the protein channel bind only to a specific molecule, such as a particular sugar or an amino acid. Once the molecule binds to the carrier protein, the protein helps or facilitates the diffusion process by changing its shape to move the molecule through the membrane and release it inside the cell. Another process is the osmosis. Osmosis is the diffusion of water across a semi-permeable membrane. Water moves from an area of lower solute concentration to an area of higher solute concentration. Active transport moves solutes and ions across the membrane against their concentration gradients. This process requires energy in the form of ATP. The best example to study the active transport is the sodium-potassium pump, or also known as sodium-potassium ATPase. The sodium-potassium ATPase exchanges 3 sodium from inside the cell to the outside with 2 potassium from outside of the cell to the inside. In this pump, 3 sodium ions bind to the inner side of the carrier protein, causing the ATPase function of the carrier protein to become activated. As a result, this carrier protein uses one molecule of ATP, releasing energy. This energy is believed to cause a conformation change in the carrier protein, releasing the sodium ions outside the cell and bringing the potassium ions into the inside of the cell. A large concentration gradient of sodium ions results due to a high concentration of sodium ions outside the cell and a lower concentration of the sodium ions inside the cell. This electrochemical gradient represents the storehouse of energy due to the excess of sodium ions outside the membrane, which is always attempting to diffuse into the cell. This active transport system may act as uniport or co-transport process. In a unitransport process, only a single molecule is transported. In co-transported processes, transport of one molecule is linked with the transport of another molecule either in the same direction, that is symport, or in opposite direction, that is antiport. Now we know that small molecules enter or leave the cell membrane through the lipid bilayer or by carrier proteins. Large molecules such as polysaccharides and sugars cross the membrane in bulk via vesicles. This process requires energy in the form of ATP. Bulk transport uses exocytosis and endocytosis for the transport of large molecules. In exocytosis, transport vesicles move to the membrane, fuse with it and release their contents. Many of the secretory cells use exocytosis for the export of their products. In endocytosis, the cell takes in macromolecules by forming vesicles from the plasma membrane. There are three types of endocytosis. One, the phagocytosis, commonly known as cellular eating. Second, pinocytosis, commonly known as cellular drinking, and the last is the receptor mediated endocytosis. In phagocytosis, a cell engulfs a particle into the vacuole and the vacuole fuses with the lysosome to digest it. In pinocytosis, the cell gulps droplets of extracellular fluid into tiny In receptor-mediated endocytosis, specific receptors embedded in the membrane bind to the ligands. From this, we can conclude that biological membranes have multiple functions, like they serve as barriers for separating the cell from its surroundings. Second, generate the intracellular compartments. Third, provide nutrient uptake and excretion of biosynthetic products. Fourth, mediate cell-to-cell communication and define cell identity. They also receive and transmit signals. With this, we come to the conclusion of this whole topic. I hope this might add to your understanding of the plasma membrane and its structure and functions. Thank you.